November StaPH-B call. I hope that you all had a great Thanksgiving, and I hope that it was socially distant. And even if you weren't able to get together with friends and family, I do hope that everybody had a chance to eat some good food. I know I did. I was actually just talking a little bit earlier. It's kind of interesting. This is my first holiday season in Virginia, and it's just kind of funny seeing what people eat in this part of the country for the holidays as opposed to everywhere else that I've lived. Collard greens and cream corn were nowhere to be found in the grocery stores. And of course I need cream corn for some of the stuff that I make. So I had to go to like 3 different grocery stores before I could finally make my Thanksgiving dinner. So that's my exciting news from Thanksgiving. Anyway, I also wanted to give a little bit of an update. There's a bunch of really cool meetings that are coming up this month, and they're not specifically StaPH-B related, but they have kind of intersecting topics, and I think that they would be interesting to the StaPH-B community. So I just wanted to mention a couple of those, just in case you guys weren't aware of them or you want to attend. So there's the nanopore community meeting is happening. Actually, I think the first day starts tomorrow. And I think it is free to register this year. It's all virtual. They did a really cool, I liked their platform for the meeting earlier this year. I think it was in June for nanopore. So that one is an interesting one, going from the 1st to the 3rd. And then there's the ASM NGS meeting, which is happening from the 7th to the 11th. And I don't know if registration is still open. I think you might have missed the discounted rate if you're signing up for that one. So if you want to go to that one, I would say either you might have to pony up a little bit more, or you might be able to sneak in with a friend or something like that. Maybe look at somebody else's screen. Shh, don't tell them I said that. And then there's the ARLN meeting, which is also going from the 7th to the 9th. So those labs who are ARLN members, they're having their meeting, and I think that it is required for some ELC kind of requirements as well. So that's another interesting one for antibiotic resistance. And I think that those were all of the updates that I wanted to get to. So I am going to go ahead and introduce our speaker for today, Dr. Eneida Hatcher. So she is going to be talking about NCBI Virus, and there's a lot of really amazing things that this platform can do, and a lot of really neat things that I think would be really interesting to our community and might be useful to you going forward in your research and in your jobs. So I'm going to go ahead and turn it over now. So Dr. Hatcher, welcome.
Thank you, and thanks, Logan, for setting this up, and everybody else for your time. So I'll be talking a little bit about NCBI Virus and the various paths that are available to getting sequence data. Oh, my thing just fell over. So I do want to say it's really interesting speaking to the public health community. I have a background in research in virus and in genomics and evolution. But it's always important for us to be able to get different perspectives from the various users that we have with different skill sets and different goals, different analyses that you're trying to do. So what I hope to do today, oh, my screen's not moving. What I hope to do today is help you understand like how to use NCBI resources to find the best viral data sets for your purposes. So I'll be presenting a little bit on virus sequence data and how it's organized at NCBI, as well as introducing our viral resources, especially those focused on SARS coronavirus 2. And I hope to have a discussion with you towards the end about a few of our future plans. So if you saw my presentation for the Spheres group, this will cover some of the same information, but it's more generally so that we can get to the discussion. So hopefully it won't be too boring for you. So let's get started with taxonomy. Taxonomy is a key attribute at NCBI. It's one of the things that links a lot of the different data types that we have. Each taxonomy group is assigned a tax ID. So for example, the tax ID for SARS coronavirus 2 is shown on the screen. Almost any of the resources at NCBI, you can use the tax ID to pull up the resources that are associated with that taxonomy group. For viruses in particular, we sync with the ICTV. So they provide the official taxonomic hierarchy and the criteria for some of the groups, but the taxonomy that they provide is the official taxonomy. And then when NCBI receives sequences through GenBank, we do the actual classification of the sequences. And that's why it's only important that they provide the criteria for some of the groups, because in a lot of the cases, we end up having to decide whether something belongs to a particular species or not. A little tip is that the taxonomy groups can occasionally shift in the hierarchy. Usually that's done to stay in sync with the ICTV, but the tax ID that's associated with a tax group should stay the same. So if you're using that particular tax ID, you don't have to worry about the shifting in the hierarchy. So I think this is kind of important. It's a little taxonomy bonus. The taxonomy browser has a lot of information that's pretty useful. So these are the two different presentations of the taxonomy browser. On the left is the hierarchy. You can search for your favorite tax group. You can change the display level so that you can see more or fewer levels of taxonomy. You can select to show data about the tax groups like the genome, and that's basically the RefSeq for that group. You can do link outs, which send you to other relevant resources, or you can see the different host types for many of the viruses. And I think if you're dealing with viruses that you're not super familiar with, it's nice to be able to see like, oh, this sequence has been found in humans, or it's related to sequences that have been found in humans. If you click on one of the tax groups, then it takes you to the page on the right. So it has more details about that tax group. And so you can see things like alternative names or the specific tax ID, or you can get links to the different NCBI databases. So on the right where it says nucleotide protein structure, et cetera. Another database you're probably familiar with is Nucleotide. So it's important to note that the sequence records here are archival. That means that they can only be changed by one of the submission authors. That means that sometimes when you find a sequence and it doesn't make you happy, you can send an email to us, but we can't actually change the information here. In the Nucleotide browser, you can do keyword searches, or you can do limited filtering with Entree search terms, or the filters on the left of the page. So I don't know if you can read it, but in the search bar here, it has TXID and then a number, and then in brackets, more information. That's an Entree search term, and you can build kind of complex searches with that. But if you're not familiar with Entree, then it's not going to do you any good. But it is very useful if you find, like there's a help document that lists out a lot of the different combinations that you can use. You can also download the data that you find in several different formats, and that's with the send to link that I have highlighted in red. But what if you only want sequences from the United States? Or if you only want sequences that are from human samples? That's where the NCBI virus resources come in handy. The underlying feature in the resources is our curation. So we use curation to mean classifying viral sequence records and normalizing the associated data like descriptors for the samples. So our curation process is partially automated and partially manual. But basically, we pick up the viral sequences really soon after they become publicly available. And then we parse the metadata from the records and normalize it into smaller but still informative dictionary of terms. And you can see an example of that in the cloud in the center of the page. We do similar processes for descriptors like host, geographic region. We try to determine if a sequence is from a lab host or a vaccine strain from the environment and it's not associated with a specific host. We also try to identify reference sequences, which we call RefSeqs for short. Those are representatives of a species, so there's usually only one per species. Occasionally there's more than one in the case of SARS coronavirus. RefSeqs can be used as a subset to measure diversity. And we tried to update significant ref-seqs with standard gene and protein annotation according to the research field. A lot of this is often done before the official taxonomy comes out. So our first resource that we started with was the influenza virus resource. This has data on influenza virus A, B, and C. It has many different filters available. And we tried to design this with the biology of the virus in mind. So you can search for the entire genome, so all the segments together, or you can search for data sets just by an individual segment. You can search for the H or the N subtype, the collection location, and including the region or as small as a state or country. I have the link for this resource at the bottom of the slide, but this will be moved into NCBI Virus eventually. Also, I didn't mention it earlier, but if anyone has questions, feel free to just pop in and stop me. So we applied a similar strategy to several other viruses. Generally, these were chosen because we had collaborators that were interested in them. So like in the case of rotavirus, or if there was an outbreak, like in the case of Zika virus. So the individual virus variation resources have, again, they're designed with the biology of the particular virus in mind. So whether it's segmented or it has mature peptides or if it has different kinds of hosts, things like that. These will also be moved into NCBI Virus. And we are hoping that's because NCBI Virus really makes it easy. This has all viral sequences. So whether it's a virus, a viroid, a satellite, any of those little weird things that we don't know how to classify, they're all there. We're providing a variety of filters that act on the normalized sequence descriptors. So you don't have to know all of the different ways of saying blood when you're searching for something. When you've selected a data set, you can easily make multiple sequence alignments, trees, or you can download the data in several different formats. We've also have interactive visualizations that help to summarize the data and help you draw conclusions for it and hopefully make it easier for you to share that information with other people. And those can be used to select the data sets. But I do want to point out that this is in NCBI Labs. So this is a new resource. It's being actively matured, it's being improved. So it might look different when you come to it from day to day, but that also means that we are really accepting information and feedback from people about what we can do to help you better. So with that, it is demo time. So I'm going to see if I can switch to the internet. There we go. So I know this is going to be a little bit small for you to read, but I just kind of want to show the layout. If you first go to NCBI Virus, so like if you do a Google search and that's how you end up here, you'll come to this main dashboard page. So at the top, there's quick links to SARS coronavirus 2 related data. There's search by sequence, which is a BLAST-like tool, and search by virus, which is a taxonomy type tool. So I'll go into those really quick. You can either paste in an accession or a sequence and do like a regular BLAST search, but when you get your results, you'll see our normalized data table. So you can continue filtering on it or just get all of the associated information in an easy way. Or you can search by a virus name or by a tax ID. We do have a few quick searches, so like all human viruses, all bacteriophages, or all SARS coronavirus 2 sequences. And again, this takes you to our results table that I'll show in just a moment. At the bottom, we have a data dashboard. So this is for all viruses. You can search for taxonomy with this sunburst. Oh, usually it looks better than that. Let's do another one. it'll take you through the different levels of taxonomy. And as you select different tax groups, this host distribution graph will change. You can also select a tax group, so let's say pig, and it will highlight the taxonomy groups that have been associated with that particular host. So this is kind of an interesting way to play with the data and get a feel for which taxonomy groups are associated with which different kinds of hosts. If you are looking for something specific, once you make your selections in both of these graphs, then you can use these statistics widgets at the top. So like if you click on all nucleotides, when we come to the results table, it'll show you all of the nucleotide records that fit those criteria that were selected. I'm going to make this a little bit bigger because this is our main feature, the results table. On the left here, you'll see we have a ton of different filters. And if you don't understand like what one of the filters are, a tooltip that pops up that gives you a definition of what it is. You can also go to our help pages, and that has more information on what an individual filter is or how it functions. We have a button for selecting columns. So some of these columns are not reflected as filters, but everything that is a filter is also a column. In the order that you see them here is the order that they'll show up in the table. We have a maximum of 200 results per page at this time. One of the questions that I have for the group though is, do you prefer seeing your entire data set at once? So like if you get 6,000 records, do you want to see all of them on one page, or is it okay to scroll through multiple pages?
Do you want answers now?
Sure, yeah.
I think for me, a lot of the times it depends on what I'm going to be doing with the data. So like if I want to download all of that, like if I can download all the records at once, that would be really nice. Whereas like if I have to, you know, scroll through each individual page and like click, then that does make things a little bit more annoying. But if there is an ability to like select all, even if you don't have all of the records on the same page, then that is nice as well.
Okay, that's a fantastic transition. So you can select all on the page with this click. It is only the things on the page though. So this is useful like if you want to do an alignment or build a phylogenetic tree. For both of those, we're limited to 500 sequences right now. Otherwise, it just is overwhelmed. And so you can use these if you only want to select like the first 200 to do an alignment. If you want to get everything, when you go to download, so let's say like you want the FASTA sequence, you have the choice of downloading only the selected records or downloading everything. And in this case, it's not limited to the page. It really is everything. So like you can download over 3 million viral records if you just wanted a bunch of data. That works the same. So we can download the FASTAs, and even if you're like on the nucleotide tab, you can download the associated CDSs or the associated proteins. You can get the accessions, or you can get the entire table. And if you do the table, again, you can do the selected records or all records, but you can choose which columns you want to show up in the table. So you can't select like what order the columns will be, but you can select what information will be in the table. Okay, I think that's everything here. Once you have sequences selected, if you either want to do a multiple sequence alignment or build a phylogenetic tree, both of them have slightly modified algorithms behind them. So they actually work pretty quickly, even if you select all 500 sequences. And now I'm going to switch to the SARS coronavirus 2 resources. So as I mentioned, there's a link at the top of the main dashboard. There's also like links from other NCBI resources, or you can Google NCBI virus SARS-CoV-2 and it'll usually take you to the right place. So we have an interactive dashboard. And with this, you can select the geographic distribution of the sequences that you're interested in, either as the countries at the world level, or you can select individual states from the United States. And we are currently adding the territories to this. So if you're interested in like Puerto Rico, it'll show up here. You can also search by the collection time of the sequences or the date when they were released in GenBank. So like, for example, where's Georgia? I just think this is kind of fun to play with sometimes. So most of the sequences that we have from Georgia were collected between February and April, so pretty early on in the outbreak. And they were released through GenBank in a pretty wide variety of time. So like seeing this as a person who works at NCBI, I would be curious if these sequences down here maybe had some trouble getting through, like getting accepted through GenBank. So I would be curious to know if like maybe they have some mutations or maybe there was some trouble with the formatting. Is there anything that we can do to make that any easier for whoever was submitting the sequences? And if it was somebody on this call, I hope I'm not calling you out. I'm not meaning to. Once you have a data set that you're interested in, and you'll see they turn gold once things are highlighted. And they'll disappear from the map, like if they'll get excluded. So if you make selections in the time sliders, you'll exclude some of the locations. But once you have your data set, you can click at the top, and this will again take you to that results table where you can either add more filtering to it depending on what you're looking for, or you can download, align, go to the associated PubMed publications, anything like that. Another thing that we have is a phylogenetic tree of complete SARS-CoV-2 sequences. This is pre-calculated, and it's pre-calculated regularly to incorporate new complete sequences. There is information on how the tree is built and what sequences are included. And
I have a quick question. When you say complete sequences, how much coverage is that? How many bases is that for a complete sequence?
So we look at between 29,600 and 31,000, but that's after trimming ends if the ends don't align with the RefSeq. Usually the ends will align with the RefSeq because they're trimmed when they come in through GenBank if they align to something else. We also look at the number of ends that are included in the sequence. So if there's a bunch of ends, it will not be included in the tree.
Okay, thank you.
Mm-hmm. With this tree, and if you build a tree in real time, you can search for any items, like any strings, any part of the string that's in the name, and you'll see it highlighted. So for example, I just searched for USA, and you can see all of the different branches of the tree that contain sequences from the USA. We're working on a better tree interaction though, so that you can search for more than one thing at a time. I think that's everything with that. Were there any questions on how NCBI Virus works, like how the table works, what kind of filtering you can apply, anything like that?
I have a quick question, I guess. On that tree of the complete sequences, is there a way you could search maybe multiple different names and have them be highlighted, or is it limited to just one at a time?
Oh, that was the wrong link. There is supposed to be a way where you can do multiple, but I don't think that it's working. Nope. We were working on it for a while where you could enter multiple, but we couldn't get it to select the colors, which seems like a silly problem, but okay. We are working on something that you can select. You can either search and color things in multiple ways, or you can select something like color by countries and then just automatically have them colored for you.
Cool, thanks.
Can you zoom in on like certain branches of the tree? Like if you clicked on a branch and you just wanted to look at those ones, could you do that?
Yes. This is another reason why we're trying to improve this. Let me see if I can click on one. Because navigating here is not as easy as it should be. So I just clicked, there's a text button at the top, so it'll zoom into the text level. Then there's several things that you can do once you're on the tree. So like you can extract a subtree. You can collapse different sections, and it has several of the details. So in the algorithm and parameters, it has what the leaf label format is, all of the information that we have in there. One of the things that I tried to do was make sure that if there's a field, but we don't have the information for that field, it's just empty so that you don't lose the counts of the fields. So it's still parsable, yes. Another nice thing is that this tree is downloadable. Any of the trees that you see like in this format, they're all downloadable. So you can get like the Newick or the Nexus and then play with it more in your favorite tree editor.
Thank you.
Okay. At the top of all of our pages is a menu. I think the most important thing here is the help document. So we have tons of information, and there's several of the items have GIFs that kind of explain it. You know, sometimes it's easier to see something, and it's harder to just read the text about it. Okay, back to the slides. So from our perspective, raw reads are becoming more and more widely recognized as an important tool in understanding the biology and the pathogenicity of viruses. So NCBI Virus gives you access to two different types of SRA data. The one in that top blue header is SRA data sets that appear to contain corona-like runs. So we have a Kamer-based tool that hopefully we'll be able to be telling you more about later, but it's a Kamer-based tool that looks for taxonomy similar sequences. And then in the table, like the results table, we have links to SRA accessions that are either linked directly to the GenBank records or that are linked to a biosample that's linked to the GenBank record. These SRA accessions are also searchable through the accession search in the filters panel. So either way, when you click one of these links, it takes you to the SRA run selector, and that has other filters that you can apply or different data that you can download. If you're interested in programmatically accessing the coronavirus data, then we have a new API. The NCBI datasets team is trying to make accessing the data this way simple. So even if you've never done it before, they're trying to provide tools to make it easier to get familiar with working with APIs. So you could go to the URL at the bottom of the slide and then scroll down the page until you see the section about viruses. And then you can click on commands to learn more about using APIs. So I thought this was kind of fun. This was really informative for me because it has like a web form. So you can build test APIs and see the results that you get. And that way, you know, like if you add it to your pipeline and it's not working, it helps you troubleshoot a little bit easier. We are also providing more resources that can be worked with in cloud environments. So right now, all short read data from SRA is in Amazon Web Services and in the Google Cloud environments. So this gets around some of the problems that are dealing with these really, really large SRA data sets. So you can use them in more computationally extensive analyses with data from more than one source. And we're also trying to provide tools in the cloud so that you can use the data and other groups can have similar processes for analyzing the data. It makes them more comparable. So these are a few of our future plans. I'm going to go over these briefly, but then I have some specific questions for you. But I'm happy to talk more about these also. So personally, I'm really interested in protein name normalization. It should make it easier to find all of the sequences that code for a specific protein. But in some cases, I struggle with identifying the most community acceptable, meaningful names for proteins. So if anybody has feedback on how to do that, I'm happy to hear it. Right now, I'm mostly working with the ICTV study groups to get feedback on names and reading a lot of papers. Our group has been talking a lot about sources for metadata other than what's already provided by sequence submitters. So for example, getting data from biosample and then normalizing that in the same way that we do the GenBank records. We are working on putting a variation tool on the SARS coronavirus 2 dashboard. So there's an example of that on the right side of the page. And again, I apologize that this is too small to really read, but at the top again, you can see the geographic map and then the collection time and the release time selection widgets. And at the bottom, an idea that we have for a variation widget. So this would show something like the number of variations across the genome. And you would be able to zoom in so that you could get a little bit more detail, and it would have more information about what are the actual mutations at that position. So if there's multiple different changes at one position, you'd be able to get more information. This is still going to be interactive with the other widgets. So for example, if you wanted to, like if you read a paper about a particular mutation, then you could select that mutation and you would see what countries it was identified in and over what time span it was identified. Or you could do the reverse. You could select a country or a state and see what mutations have been seen in that country or state. Obviously, this is limited to the sequences that have been submitted to GenBank or to the INSD collaboration. So I have a few questions for the audience. I hope you don't mind. Also, I'm sure there's some people in the audience who prefer to contribute quietly because that's usually how I am. So please respond in the chat or send me an email, and I have my email at the end of the slides if that's how you prefer to send information. But I was curious though, in normal years, so not SARS coronavirus 2 years, is most of your work virus or bacteria? Because it seems to be maybe more heavily bacteria. But if you do work often with viruses, I'm curious to know which viruses. And again, you guys can just shout it out or you can add it in the comments.
I would say that normally we typically, at least from my experience, are more bacterial focused and the types of organisms that we're working with. And for viruses, I mean, there's kind of like some of the big ones that are important to public health, I think, that people work with. So there's like flu, noro, HCV, those kind of things. But anybody else is welcome to shout out and kind of give their answers too. I don't want to hog the conversation.
I'd say for us in Minnesota, at least, it's very similar to what Logan said, and then add in some of like the vaccine preventable viruses like measles, mumps, rubella, that kind of thing.
That's good. I was working with a group on some measles stuff, so it's good to know. Norovirus is also one of the viruses that we've done something like with SARS coronavirus, where the annotation is automatically applied when you submit to GenBank. So hopefully norovirus, and I'm hoping that we can get measles included in that group sometime soon, because I've already done some testing with it.
There are a lot of people doing like, rabies sequencing too? Because I know that that was something that I kind of talked about and just kind of popped into my head.
Sorry, which one?
Rabies?
Oh,
I wasn't sure if people were doing that widely or if that was just kind of like a fringe thing.
I'd say, yeah,
I see it a lot on the ProMed Health.
Here in Massachusetts, we have done some rabies. We also were looking at trying to do West Nile and Tripoli, though COVID took over everything. So maybe someday. We definitely do some Hep A and have thoughts of trying to bring TB on, but that's kind of a big lift. I know there actually are some other labs looking at that as well, but that's not really a virus for you.
Okay. Also in normal years, because I've had some, that measles project that I mentioned, it sounded like the group who was interested in working on them was having some trouble knowing like what they were submitting to GenBank was the right thing to submit. The virus group is always happy. Like obviously we are not GenBank, we don't accept your sequences, but if you have questions about annotation or about like how you can find sequence sets that you're looking for, we're always happy to help you with looking for that kind of data. So, okay, next question. And so I've seen some talk recently about getting data from either GenBank or from NCBI Virus and then adding it to other epidemiological tools such as MicroReact or NextStrain Augur. And it seems like the biggest problem with that is having the sequence ID match between the FASTA and the metadata table that you have to download. So I thought that the easiest solution for that would be if you had like a toggle, an option to get you to the accession.version or just the accession. So if you see accession.version, it'll say like letter, number, whatever, and then it'll have a period followed by like 1, 2, 3, something like that. For us, that means a lot. But maybe for the community, having that version on there is not really useful. So would you prefer having something like a toggle where you could turn it on or off, or just not seeing the version on there at all? And I can give examples if that's more helpful.
Yeah, I think I'm a little bit confused. So what's the version number we're talking about when we're getting the accessions? Just like, I'm not sure.
Yeah, I'll pull it up on I'll pull up an example. So when you see records like this is just the accession, but these are often followed by .1 or .2. So like if you downloaded these sequences as FASTAs, it would have this number .1. And that's sort of, it's a slightly different format than what you would get if you just like downloaded the table. And that causes problems with tools like MicroReact or like NextStrain where the sequence IDs have to match exactly. So as I mentioned, I think when I've talked to other people in the community, seeing the sequences like this without the .1 is perfectly fine. But I just wanted to double check. That's not useful to anyone. What it means, so if you see a .2 on there, it means that the actual sequence, so like the nucleotide sequence, has been changed by one of the submitters. So like maybe there was a gap that they were able to fill in, and then it would go from .1 to .2.
So yeah, I guess just for us, as long as the title matches whatever kind of like sequence we're downloading. So like if the name of it matches the name in the FASTA file or something like that, or FASTQ file, then that is better for us, I think. I mean, I don't think it normally really matters, but yeah, if we are going to be using it for like a NextStrain or kind of putting it into these other programs where it is important to have the names match, then yeah, that does get to be a headache if you have to go through and change everything. So if you have a way to do that automatically, that saves us time, I think.
Yeah, it's a little bit of work on our side, but I think that consistency would be really useful on your side. That's my impression. Okay. I mentioned this earlier, we are currently limiting the multiple sequence alignment and trees to 500 sequences. So this does not apply to that pre-calculated SARS coronavirus tree. That one has as many as needs to be in there. But if you're doing if you're selecting sequences and making a real-time tree, it's limited to 500 sequences. Is that generally okay? And if you wanted more sequences, what are you trying to learn from that kind of tree? Or really, if you're making trees at all, what are you trying to learn from that tree? While you're thinking, I'll admit, this is one of those places where I'm really biased because I've been making trees for a long time. So I know what questions I'm answering, but I want to make sure they're the same questions that you're trying to answer.
I mean, I think from our end, when we're making trees with 500 sequences, we'd probably be looking at kind of geographic regions and trying to see if there's, if there's kind of distinct clades or kind of, differences that we're seeing between different populations that we would, be looking at kind of either in a surveillance capacity or if we're actually looking at across state lines or something like that. I think that's typically where I would use more than 500 sequences. Otherwise, typically we're not making trees that are quite that big. We're looking at more kind of I don't know, drilling down into specific outbreak scenarios and things like that, where we have a limited number of sequences, and then we're trying to see if they're all related to each other. Luckily, I haven't seen any outbreaks yet where it's been over 500, people who were involved. But if there's any other things that people want to add, I'm all ears.
So that's good to know. So it seems like maybe for your purposes, trees that show more of the individual mutations from one part of the tree to a different part of the tree might be more informative. Okay, that's good. Okay, specifically related to SARS coronavirus 2. Are you using lineage information? If you are you using pangolin? The DoD has a lineage proposal. There's a few others that are out there. Do you use this for selecting sequences or for tracking diversity, or are you using individual mutations for tracking diversity? Or has this not really come up for public health groups in general?
I know personally here, we like to use the pangolin lineages. We found them to be kind of helpful when we're tracking diversity. Sometimes the individual mutations can get to be a little bit kind of too much. And so it's nicer to have those kind of, more zoomed out filters that you can kind of apply, the pangolin lineage or the next strain clades and those kind of things. So then they can help us kind of sort through things and look at more of a general picture. I haven't used the DoD lineages yet, but that's something that we might look into.
Okay, so it's kind of more about giving it a name that you can talk about. Okay, cool. And this applies to bacteria and to viruses. Do you usually work with whole genomes or with individual proteins or both?
I generally work with whole genomes, although we have been working on a project where specifically we were looking at sequences of the spike protein.
Okay.
Yeah, I think generally for most people, we work with the whole genomes, but then if there's like a specific individual protein of interest that jumps out in the literature is like important to something, then people will look at individuals, but for the most part, it's more the whole genome approach.
Okay, good. And this last one I hope is kind of a fun question. So my understanding is that if you're working in public health, you often have to present your data, present your conclusions to your coworkers. So that might be clinicians, epidemiologists, policymakers. Please let me know if I'm missing or just wrong on any of this. So for example, if you could select a sequence and then see it as a 3D molecule with the variations highlighted, is that an easier way to present your conclusions to somebody else to help them understand the importance? And so I have an image of what that might look like. So like this is the wild type spike protein, and in the center, you can see the amino acid that changes, and you can see how it's connected to the surrounding molecule. And then in the mutation, please ignore the background coloring that just changes. We're only focusing on the center. You see that you lose a lot of the connections. So you can show something about the significance of a single amino acid change as a picture, which might be more impactful. But this is all just me assuming things. So if you have ideas about visualizations that would be easier for you to understand or for you to help others understand what you're seeing, we're really interested in helping you produce those. So the way that we see it, it's probably easier for us to allow you to make selections and then see the data how you want to see it, and have it kind of a uniform process that can be shared with other people, instead of you having to learn a whole other program or visualization or something so that you can make those things yourself.
Yeah, I think that that's really cool. I like that a lot. And if it's easy to use and people can use it pretty effectively, then I think that it would make a difference, at least for our purposes. And I know that we just like to kind of like look at those things sometimes just ourselves to see like, okay, well, you know, if we see something in our data, does it look like that would, you know, really affect the way that the protein is folding, or would that, you know, have any sort of kind of like implication in sort of like an active site or something like that? So if you have a way to make that happen easily, then yeah, I think we would use that. I think a lot of us have done that.
Okay, good. It's one of the, there's a tool at NCBI, it's linked here at the bottom. It's called IC in 3D. And I've been playing with it a lot lately. And I'm really hoping that we can connect IC in 3D to more of our resources, because I do think it's really important, right? You can see a sequence that has a bunch of mutations in it, and it might not mean anything. Or you can see a sequence that has one mutation in it. And if it's in an active site, that can mean a huge change to the biology. So at least from my perspective, I think that's something that's really informative, but I want to make sure that people are interested in using it. So I did list a whole bunch of links here for reference. I'm assuming that, I know that the video will be shared, but I'll make the slides available to whoever wants them. So there's a bunch of different links here if anybody's interested in seeing any of the resources I talked about today. I also listed some of our cloud inventory if anybody's interested in playing with cloud data, and highlighted some ways that you can find help on our resource. And I wanted to thank everybody for your attention. My email is here on the slide. Feel free to contact me about anything in the presentation, or if you just have general, how do I find this, or what does this mean kind of stuff. I'm always happy to talk to people, unfortunately. I think that's it. I also, at the end of the slides, I have a bunch of like extra, like an appendix, just for more information if anybody is confused about how to find something. It's all there.
I think that this was fantastic. It was a great talk, and look at that, we were right on time too. We have a few minutes now. I think if people want to ask questions, I have a few. So, but I've been talking a lot, so I feel like I'll give other people a chance to ask questions, and then I don't know. Also, if you're not comfortable talking, we can add them in the chat too. I'll give people a second. Well, if nobody else has questions yet, or if they think of them as we're going, I had a couple of questions. So with all the resources that you have available on NCBI Virus, is it only, can we only use data which is already on NCBI, or if we have like a sequence ourselves, could we like upload that to some sort of, I don't know, part of the website and then like compare that to other sequences that are already there?
Right now, you can only do the sequences that are part of the data set. One of the changes that we're hoping to make in the next year or so is incorporating more community data. So whether that means like somebody has a table of research that they've done and they want to share that with other people, or that means just uploading an individual sequence and seeing how it fits into a phylogenetic tree, which I think is probably the most applicable way of uploading a SQL sequence, is either a MSA or a tree. So yeah, that's something that we're thinking about. Yeah. Okay.
Yeah, that's really interesting. And then I don't know if you are the person to answer this question, but I'm just wondering, do you know if there's going to be more RefSeq sequences for like coronavirus, or are they just going to keep the one? Because it seems like as you know the virus kind of continues and mutates a little bit, the original RefSeq sequence may not be as, I don't know, kind of relevant.
Yeah, the sequences are moving away from it, right? Mm-hmm. So, okay. Usually what we do is one RefSeq per species, which is SARS coronavirus. So we already have two. That's not a problem to add a third one. What I have not been able to do is spend very much time identifying what that third one would be, right? So what I could do is pick a sequence that happened relatively soon after the split, the D614G, I think, after that split, but that happened pretty early on. So I don't know if that sequence would be representative. So I would just have to find, and if anybody has any suggestions of what another RefSeq would be, I'm the one who creates it. So email me. And the same applies for others. So like Coronaviridae in general, maybe not right now, but probably in the future, there's going to be more research about like the entire genus or the entire family. And so I've also been trying to look at the related RefSeqs and make sure that they have appropriate annotation and that we have good coverage for the diversity that's in the groups.
Thank you for that. All right, it does look like we are at the top of the hour, so I want to let people know if they have other things that they have to get to. And then if not, I think I have a few more questions, but they're not really as related to this. So I just wanted to let people know that it is the top of the hour, and you're welcome to leave if you want. But if you have a minute or two more, Eneida,
yeah, I can stick around for a while. I have 30 minutes.
Okay. Yeah, I don't think it's going to take that long. And we've already taken up so much of your time. But I just had a question, because in the beginning of the presentation, you were talking about the taxonomies, and then you said that sometimes they change. What would cause a taxonomy to change? This is just kind of my own personal curiosity.
Yeah. Okay, so I'll give you an example that's sort of related to the virus of the day. So as I mentioned, SARS coronavirus is a species. And then underneath of that, there's some bat SARS coronavirus-like viruses. And then there's viruses from the original outbreak in like 2003. And then there's also the modern SARS-2. So people were having trouble identifying sequences, like separating them out from the original pandemic, as opposed to like the bat and pangolin and other animal coronaviruses. So in that case, we shifted some of the sequences so that if we could tell that it was from the original pandemic, we moved it under a group called SARS-TOR2. So like the same way, like we have WHHU1 for Wuhan human 1 for the early name of SARS-2. It was the 2003 pandemic was called TOR2. So like in that case, the tax group didn't change, like the number for those didn't change, but the individual name changed. And they're still underneath of the same species. So we'll have, or sometimes like another example is there will be a sequence that is unclassified. So it's assigned to a family, but it's unclassified other than that. So no genus, no species. And then at some point, there will be a taxonomy update, or we'll get more viruses, something like that. And we'll see that that unclassified sequence is similar to another group. So we'll shift those underneath of the other classification so that they still have their same name, because if you're reading a paper and you want to look for something, you're going to use the name that's in the paper, and that's not going to change. So yeah, that was a confusing explanation, but taxonomy is confusing.
No, yeah. It's, you know, a man-made application to something that happens in nature. So yeah, I think that was a good explanation. Thank you.
Yeah.
And then
it's one of the reasons that I like the taxonomy browser. Sorry. It's because like with the taxonomy browser, you can visualize the hierarchy, which is sometimes important if you're not familiar with a particular virus group. It kind of gives you a general picture of who's related to who. Mm-hmm.
Yeah, and I did think that it was interesting too, like when you highlight the different hosts and stuff like that, you know, you get these light ups on that kind of like graph of which viruses infect those things and then where they kind of fall, and that was interesting, I thought. And then my last question that I have is there an explanation somewhere of the kind of back end of what you're using to like build the multi-sequence alignments and the phylogenetic trees? Like, do you have a kind of like step-by-step outlining, you know, which programs you used or what kind of parameters and things like that?
It's in one of our publications, and I have to find it and add it to the help document. So the alignment is a variation of muscle that one of our developers like tweaked the algorithm for muscle to get it to work faster. And then the tree is based off of the same alignment. And I think that it's a neighbor joining tree, but I have to double check that.
Okay. Interesting.
Yeah. So I was kind of a snob when I first started about like the way that trees are built. Like I really wanted my Mr. Bayes phylogenetic trees and hand editing my multiple sequence alignments. So it took a while to convince me that the alignments in the trees that we were producing were worthwhile because we were making them so fast. Mm-hmm. And there are times where the tree will change, like it'll be different from like my very long process tree. But in general, the topology is very, very similar. So you'll have, you know, within branches, like things that are very closely related to each other will often like flip-flop when I would compare the two. But in general, they're actually really close.
Well, that's really interesting. And it's a good kind of, I guess, confirmation that the processes, the speed ups are actually worthwhile and they're working.
Yeah.
Well, that's interesting. Neat. All right, well, that was all of the questions that I had for this. I thought that this was a really cool presentation. I'm excited to kind of like play around with NCBI Virus a little bit more. And I mean, I think we have been kind of exposed to viruses a lot more because of SARS-CoV-2, but I'm interested to see if this will kind of spur some more interest in other viruses as well, and if we'll start kind of looking at some other things. I know some other people, it sounded like, had kind of different viruses that were on the docket kind of before SARS took over everything. So this is a good resource, I think, for all of us to have kind of in our little toolkit.
Thank you. So just to reiterate, if anybody has like projects that they're thinking about starting and they have questions, so maybe you want to know like what kind of resources we have available for measles or for rabies, or you want to know like, can we do annotation on those sequences, then please contact us, because the way that we try to work is making collaborations with people outside of NCBI. And that way we have somebody to make sure that we are working in the right direction. So that's why we keep asking for people to reach out to us.
Good to know. Are you using, is it Vader for the annotations that you're doing?
Yeah, that's,
Okay.
I helped develop Vader, and that's the one that, so we're using it now for norovirus, dengue virus, the SARS-CoV-2. There's a few other viruses that are not, like, it's been tested on almost all the Flaviviridae, but it's not in the GenBank like acceptance pipeline yet.
Okay.
But if you're, sequencing a whole bunch of West Nile virus and you want to annotate them, then we can send you the right Vader tool for it.
Well, that's good to know.
Well, I really appreciate everybody's attention and the questions and everything. Thank you.
Thank you. Thank you so much for agreeing to be on the call. I was really excited to have you on, and so I'm really glad that we found a chance to snag you for a little bit and it was like, ask you questions and see this in person. And that was very brave of doing a live demo, but it worked flawlessly. So if there's any other questions, speak now, or we have
Or send an email.
yeah, email right here, I was going to say. No need to hold your peace. But yeah, thank you so much again, and we'll hopefully continue to stay in touch.
Great, thanks. Everybody have a good afternoon, depending on where you are in the world.
All right, bye-bye, everybody. Oh, well, just as a reminder, the video will be up on the website after it's finished rendering, but I'll send out an email with the link, and I'll include Eneida's information in the link as well, just in case you want to send her any more questions.